I wanted to welcome everyone to this session this afternoon on to learn more about COVID-19. And I am the chairman of EPPIC Global. A little bit about EPPIC Global. It's an organization that was founded by like-minded people in the Bay Area for networking, mentorship, and entrepreneurial support for people in the healthcare and life sciences industry. We have had a lot of members who are our core members who have stayed on with us for a while. And we have also started adding members who have more medtech and digital health based expertise. So welcome to this session today. We will have this session with great speakers. Sanjeev Redkar will start out by telling us about this enemy that we are facing today, COVID-19. Next, Saroja Ramanujan will talk a little bit about COVID modeling. We've been hearing a lot about this in the press. So she'll go through some of the modeling and the assumptions and implications of those models. And then we will hold any questions that people have till the very end, where Anurag Mairal will moderate a panel discussion, and he will capture all the questions that people have. You can just add your questions in the chat and we'll keep track of it. And at the end, Robert DeFeo, the president of EPPIC, will tell us a little bit more about EPPIC, and we hope everybody can stay on till that session because we have some really exciting news to share with you. EPPIC has been revitalizing during this period of shelter in place. So with that, let me hand over to Sanjeev Redkar.
Great. Hey, hello everyone. I see now we have 22 participants. Yes, 16 plus, which is good. Hello everyone. Good afternoon. Let me share my screen. Let me see here. Are you able to see the screen?
Yes. Yes.
Wonderful. Wonderful. Well, let me start out by, this is a short slide deck. It kind of touches on a few different things, but I want to start out by saying I'm by no means a virologist, epidemiologist, or an expert in this field, but just like you all, we are all voraciously hungry for this information. And, you know, I was thinking as I look at, you know, this EPPIC family, we are now 25 of us on this call, and there are a lot more. And this is just at a short notice of, you know, 2 days, 25 people are joining in. What is our responsibility as scientists? Somebody that has been in the industry for so long. I get a lot of questions. I think I put it in 3 bullet points. One is we will help our colleagues, friends, family, help understand the science behind what we are seeing, what is happening. So much data, we're getting tremendous data overload. Try to at least make sense out of these data and really reassure everyone that science will indeed triumph in the end. And I'm tremendously confident in the community as well as our colleagues. So let's see, why is this strain so infectious, right? We know that we have had the SARS-CoV in the first time in 2003. About 80 to 86% of the genome is shared, but look at the difference in the infections and death. Only 8,000 infections and 774 deaths in 8 months, whereas SARS-CoV now has greater than 2 million infections and 150,000 deaths now in just barely 4 months. And the key really what immediately protein biologists did, identify it's the spike protein on this new coronavirus, which has now a 10-fold greater affinity to the ACE2 receptor on type 2 human cells. And this is a key receptor in a lot of endothelium. And the second differentiator is that this virus blocks interferon. It remains dormant for a certain period of time before symptoms start reappearing. You know, we have seen that the primary organ that it infects is the lung. In a healthy lung, you've got type 1 alveolar cell, type 2 alveolar cells. The type 1s are the small cells where the oxygen and carbon dioxide exchange takes place between the vasculature and the alveoli. Type two cells are little larger cells, and their responsibility is to coat the walls with a surfactant-like substance. And what they are, the cells are high expressors of these ACE2 receptor. This is the entryway where the RNA, the viral entry point into these large cells. And what happens is that once it enters the cell, replicates, destroys the cells, and continues to infect more cells. And then the patient starts getting COVID-19. That's the disease caused by the SARS-CoV-2. The moderate disease is when you start getting an inflammatory signal where these now all released cell debris causes an inflammatory response. Macrophages, cytokines, fluids start building up in the alveoli, and now they have hard of breathing. Oxygen levels go down. Severe COVID is where you have complete blockage. Protein-rich fluid is in there, and this is when the patient has to be intubated. So almost these patients fall into these two categories. Moderate categories where they may need oxygen, and severe is where intubation is needed. What is even more interesting, this is a little busy slide, but this COVID-19 is distinct from any coronaviruses before. This is a paper that was published yesterday in Science, a few days ago, yesterday, I believe, in Science. On the left-hand side, it tracks symptoms. We're finding now anosmia, loss of smell and taste, as a sign. A large number of patients. On the left side, what you see is you see a map of symptoms of these patients. Now, this is through an app, so it's not 100% accurate. But to the right, what doctors are finding is that patients are not dying just because of the lung issue. Now, this is not just ARDS. Patients are dying because of clots, coagulopathies, kidney breakdown. They see extreme diarrhea, nasal heart attack is a common occurrence, stroke, pulmonary embolism. So this is a very different disease. Today there was a paper that was published. There is a marker called D-dimer. It relates to whether you've had a stroke, fibrin related, and you see that patients that have the D-dimer stay in the hospital longer. So this is something that doctors are tracking and will know a little more in the coming months as doctors have more patients to care in COVID-19. The next section, let's talk about testing. We've heard a lot about testing. There are two types of tests, molecular and serology. Molecular are detect the SARS-CoV-2 RNA. It's a nasal swab. It can be a point of care system, uses the RT-PCR assay. Now FDA, none of the assays are technically approved, but FDA has emergency use authorization for about 35 companies, including, CDC was the first one, Roche, Thermo Fisher. The good part is they have high accuracy, sensitivity, as well as specificity. Also remember, it's a qualitative test, and the LOD is about 10 square copies per ml. What a lot of people are now talking about is a serology test, which is basically now have these patients developed antibodies against the virus. And there are now 4 antibody tests that have received emergency use authorization. Several others are being developed. Roche just announced that they have one. They will file to the FDA. This is also a qualitative detection. However, this is an ELISA test. The body develops IgM when you have recent exposure, and they develop IgG, prolonged later exposure. This is a much faster test. But understand that for these two tests, for serology tests, you want 0 false positives. Whereas for a molecular test, you want 0 false negatives, because you don't want anybody that have the disease that you're not able to detect. So they have different criteria. So on this slide, I just took the summary from the first serology test that FDA gave a EUA, the SELEX test. Now, as you can see, In this test, they do something called a positive percent agreement and a negative percent agreement. What that means is that it has 4% false positives. The negative percent agreements means 240 out of 250 were real negatives, and the other 10 were false positives, which means they were positives that were that incorrectly came as negative. So now let's take this test, right? Let's see if I take this 4% false positive rate and I implement this test on a random sample of 1 million population. To the left, if you see random 1 million population, and let's say the incidence is 1%. So there are 990 that are not infected. Because the false positive rate is 4%, I almost have 44,000 or 40,000 false positives. If you actually go through the math, if you utilize this test in the public in a 1 million population, 82% of these tests are going to be false positives. People don't realize the importance of 0% false positives. So we are seeing a lot of these tests that are not FDA approved. Papers are going to come out. I think devil's in the details. Now, Ortho's test has a EUA, has a 0% false positive. And I'm sure Roche, when they give out information, knowing Roche, I'm sure that will also have 0% false positives. And those are the tests that we'll have to utilize. But I just wanted to bring to your attention the difference between these molecular and serology tests. Let's move on now to therapeutics and vaccines. That's the next section I want to share from the reading that I've done. But before we go there, understand that there are 4 distinct populations in this COVID segment that we need to cater to. Of course, first one is the general population, which needs a prophylactic vaccine that would be very long-term. These takes 4, 5 years, where you absolutely not only see the increase in antibody titer, but you want to see long-term immunity. Then those that are tested positive, therapeutic or a vaccine. And then the 2 categories I told about, whether they're moderate or severe COVID-19. One that are on full oxygen support and others that are not. And WHO now has given a measure for success. It's an odds ratio on a 7-point scale. So NIH is also having a harmonization between the clinical outcomes. So these trials can, especially the randomized control trials, can utilize the same criteria. Before we go into therapeutics, what was amazing is immediately right in January, the sequence for SARS-CoV was determined by Chinese scientists. And now recently in March, the exact region, the receptor binding where the spike protein of the SARS-CoV-2 binds to ACE2 has been identified. And what you see is that it's a very small 120 amino acid region. The one to the bottom left, those red that you see, those are the differences between SARS-CoV-2 that had such a high infective rate and SARS-CoV, the receptor binding region. And there are very small, the black dots where they bind to ACE2 versus the red dots. And what is amazing is just this small difference causes such a difference in fatality and in infectivity. But this was important for scientists to provide, this was the first step towards making vaccines. But before we go into vaccines, I want to talk a little bit about therapeutics. There are 7 areas of therapeutics that we are working on. One are the nucleoside analogs, remdesivir. This is Gilead's drug. It's an adenosine analog. It gets incorporated in the RNA termination. Now, the good part is remdesivir was being tested. It was shown, it was already in the clinic. It's not approved, but it has shown activity against the SARS-CoV, the first one, as well as MERS in animal models. And it works against Ebola. So they had, so they were a step ahead of others. The others are protease inhibitors. Abbott has 2 of these approved for HIV. The third is a very interesting, because of the immune reaction that I showed you, the cytokine release arm, and now we are learning more and more about different organs. IL-6 antagonists that are approved for rheumatoid arthritis. they have shown some early activity, and a lot of trials are trying to see whether IL-6 in early or late patients provide benefit. Hydroxychloroquine, azithromycin, that's also another one. It's an interesting story. I'll have a slide on it. And then there are a few others that use convalescent serum, hyperimmune globulins, which are high titer neutralizing antibodies, as well as recombinant human ACE2. Now, remdesivir had some very good compassionate trial data. This is a single arm trial, 53 patients. But what they showed is here, if you see that graph, you can see patients that are out of the 50 patient who were invasive in the top row, how many were non-invasive, and once they took the drug, it's an IV drug, how many were converted from one bucket to the other. So how many invasive became non-invasive, how many non-invasive became low flow oxygen. Basically, fatality rate in this trial was 13%. Normally they would expect 50% fatality. These are very severe patients. 34 of them were invasive, which means they were intubated. This is certainly a positive outcome, but a lot of people are saying without randomized controlled trials, it's very hard to know whether this is indeed better than the standard of care. Gilead is already doing 2 trials, one trial in severe patients, another in moderate. They increased the number of patients from 400 to 2,400, 600 to 1,600, and the endpoint is also changed. Now the 2 RCTs from China were closed because they could not recruit. So it's very hard to what to make of this. Only time will tell how these 2 major trials. But I give Gilead credit for putting a lot of effort moving this along. Protease inhibitors, the lopinavir, ritonavir, darunavir from AbbVie and Janssen, you know, they're taking a hands-off approach, both companies. They're giving as much drug to support a lot of RCTs that cooperative groups are doing, institutions are doing, but they're both are saying that they don't have access to the China trial data, and their main priority is to make sure that the supply chain for HIV is not disrupted. But they're giving and supporting the trials that are going on in COVID-19. Anti-IL-6, 2 of them are approved, Sanofi Regeneron's Kevzara and Roche's Actemra. Tocilizumab, Actemra had some positive results coming out of China that showed that 15 out of 20 patients reduced their supplemental oxygen after receiving Actemra. So these trials need to prove, because IL-6 is clearly a prognostic marker of severity. The question that we don't know is the cytokine by blocking IL-6, because you also want the immune system to fight the disease. So this balance of fighting the disease versus blocking the cytokine storm, this remains to be proven. But Roche and Sanofi Regeneron both are very gung-ho, have started randomized controlled trials in this setting. And in the clinic, docs are already using it when they see signs of CRS in these patients. Now, HCQ, azithromycin, we have heard a lot. It is a drug approved for malaria, RA, lupus at a lower dose. What was found, a Nature paper, that in vitro, they were able to block the transport of SARS-CoV-2 that caused a lot of excitement, except that it was at very high concentration, 1 microgram per ml. That concentration is not reached with the malaria dose. So Dr. Didier Raoult, there's a French doc, he published a couple of papers. The second one has been now retracted. What they showed is they showed faster elimination of virus by PCR in first 36 patients and then 80 patients. The 80 patient paper is now retracted. The levels reached were only 0.2 to 0.4 microgram per ml. Subsequent 2 RCTs, a French study and a China open label study, control trial, the 2-arm open label study, showed no difference between HCQ and no HCQ. So this may have a place, maybe in a prophylaxis setting. There are RCTs going on in high exposure environment. These are healthcare workers that are taking care of patients. Only time will, because that's how the drug is used in malaria. It's given at a higher dose, 600 or 800 milligram, and that's where it's being tested. Now let's go in the last few about vaccines. Now, the key to the vaccine is that spike protein that I mentioned to you. The spike protein, the antigenic region where it binds to ACE2, that's the key that everyone is going after. Here on the slide, you can see different types of vaccines. There are RNA vaccines, which are basically, these are RNA that are encapsulated. There are two companies that are in the front running, Moderna and Pfizer-BioNTech. They are in the running for RNA vaccines. You can have DNA vaccines. So this could have a vaccine that is a DNA plasmid that enters into the cell. And all of these, the job of all of these is enter into the cell and spit out the antigenic portion of the spike protein to start the immune response in the body and develop the neutralizing antibodies. The third bucket are recombinant protein vaccines. Understand that RNA or DNA vaccines have not been approved yet for any infectious disease. Recombinant protein vaccine is a very common one. Influenza, several have been approved. GSK, Novavax, Sanofi, they're all working in this area. And then the others are vectored vaccines, inactivated vaccines. Vectored vaccines means you put a DNA or RNA inside a viral vector. And there are several companies from CanSino. They started phase 2 working in this. I want to mention Serum Institute of India. I was looking them up. They have a live attenuated vaccine, and it's about, this company is owned by, Cyrus Poonawalla and from Pune. And these guys have a live attenuated vaccine. And what was very interesting is his son, Adar Poonawalla, runs this. He said that he wants to not file a patent on this, and he wants to develop this vaccine, and they're looking at going into the clinic. They've started animal studies. Late in the year, they're working with Codogenics from US. They're our largest manufacturer of vaccines in India and supplier of a lot of vaccines in all over the world. And it's something that we could follow. So there are now 5 vaccines in the clinic already, and that's amazing. Moderna's LNP, so it's a liposomal encapsulated mRNA vaccine that's in phase 2 that has just started. CanSino Biologics, they have adenovector type 5 vector that expresses the S protein. They have started phase 2. They finished phase 1. They're ahead, of course, because they're a China company. Inovio is the third one that has a DNA vaccine, a plasmid. And then there are 2 other cell-based vaccines that I couldn't find much information on. So in the next few slides, I have a little bit of information on the first 3. Moderna, they're working with an IAID. They don't have any mRNA vaccine approved yet, but they have a vaccine in CMV that has shown positive data in phase 2. Phase 3 is to start in 2021. They have other programs also early, but they've already begun this, this phase 1. It's in healthy volunteers. They are April 8, they started the highest dose, 250 microgram. And theirs is a shorter readout. Their primary endpoint is basically safety, and the secondary endpoint is immunogenicity. So they're looking at antibody production at day 57. So this readout should be pretty relatively quick, whether this approach is indeed working to develop antibodies. And they have also committed to delivering a certain amount of vaccines by the end of the year, doses, I mean. Inovio is a company that has been in the business for a long time. Now, think of it like this. The previous one was lipid encapsulated, so they could be taken up by cells. This is a naked plasmid DNA. Now, this is not taken up by cells. So Inovio has a very interesting way. They have a device that is electroporation device that enters intradermally, gives out electrical pulses, breaks open the cell, the plasmid gets incorporated. incorporated into the cell, and then it elicits a dendritic cell response, adaptive immunity. Here they have an HPV vaccine that is in phase 3, sorry. Again, no approved programs at all yet. But Inovio, they have started phase 1. They're going to start phase 1 in April. So very soon they'll start their phase 1. Oh no, they're already in phase 1. Yes, that's right. They started in April. That's right. CanSino, they're using a replication defective adenovirus vector, type 5 vector. This vector expresses the spike protein. Not much is known, but their non-clinical data, they have said that they saw in non-human primates the antibody titer, and they have started phase 2, but all their trials are, the sites are in China. J&J is another company. They have a platform which is similar to adenovirus vector platform called 8026. So this is also a replication defective vector. They used it for their Ebola vaccine. Now, Janssen has 12 clinical studies. It's not yet approved, but they have applied for market authorization in Europe. So you can say that it's a tested platform for Ebola. Merck got its vaccine approved. I am presuming that Janssen will also get the Ebola vaccine approved. And they've committed to using that platform for SARS-CoV. However, they've said that by September, they will start their first in-human study from a manufacturing perspective. But knowing their vaccine progress, once they come out, their manufacturing and supply chain, I believe, will be rock solid. You all know about the Sanofi-GSK collaboration. Both are behemoths in vaccine. Both have tremendous collaborations independently to each other, and they're working with CEPI. GSK is working with CEPI, Clover. And here what they're doing is, I forgot to mention, an adjuvant is also equally important. And GSK is going to bring its adjuvant technology. Sanofi will bring the protein-based vaccine, which will be an antigenic segment of the spike protein, and they will work together on all these partnerships to develop, work with NIH to initiate a program of vaccine. Normally it would take 5 to 6 years, but I trust that we should have something next year, in my opinion, based on how things are going. You know, this concludes my presentation, and I'll happy to hand it over to Saroja.
Hi, so are we waiting, holding questions till the panel?
Yes.
Yeah, what we can do is that if people have questions, oh, they're already on chat. Okay, great. Okay, wonderful.
Yeah, I've been gathering questions, so we'll jump into it as soon as Saroja is done.
Let me get my, how's that? Do you guys see this? Hello?
Yeah, we can see it.
Yes.
Okay, wonderful. So you just heard a really nice overview of the biology of the virus, how it infects, how it causes symptoms, and the therapeutic avenues that are being explored to address it. And actually, that's the world I really live in day to day, is more in the drug development space. And that's where I'm a computational modeler and a drug developer, especially preclinical, translational, trying to make sense of that. However, with all of the modeling, the epidemiological modeling being presented recently in the news and discussions of flattening the curve, etc., I started conversing with many of my colleagues who were asking me questions about these models, and the methodologies are very similar. So I thought this presentation of the different approaches that are being used in different contexts for modeling the epidemiology and the spread of COVID, the assumptions behind them and the implications and the projections of these different models are very important for us, even as lay people, to understand. There we go. Okay, so briefly, these highly cited and presented COVID models are driving the public discourse. So I think it's important for us to understand what data they're built off, what their strengths, and what the caveats are. I'm going to go through a few different very influential models and the different modeling approaches at a very qualitative level, not a lot of math here or anything, so that people understand when they see them. Disclaimers, I am not going to include a lot of maths because this is really for all of us as general scientists or lay people even to understand. And as I said, I am not an epidemiological modeling expert, so please take all of that into account. And as I go through, I'm also going to highlight some quotes from a Nature News feature special report called The Simulations Driving the World's Response to COVID. You'll see those in this kind of yellow or kind of peach colored boxes as we go. So the first part of my talk is about early on in the COVID infection as the data comes out. How did we understand the dynamics and how was modeling used to understand that dynamic and the risk? And I think there's various events that happened early. You guys also probably heard about this R0, which is the idea of how many people does one infected person subsequently infect. It's a measure of the transmissibility and the spread of the virus. So some of the very standard or workhorse modeling approaches used here are based on the idea of lumping your population into different states. So you have the susceptible and the exposed. Sometimes these are split out, sometimes they're together. And this, if I tie back to the previous talk, are the patients who would have neither tested positive for the COVID RNA, nor would they have tested positive on serology for the antibody. However, over time, the infected individuals will come back and will convert susceptible people to exposed people, and they may become infectious over time. And over time, the infectious people will either pass away or they will recover. And at that point, they could be negative for the actual COVID RNA, but they would be positive on serology, hopefully. So this is how I tie that back to that testing. The thing about these lumped models is they don't consider different subpopulations within these typically, unless you break them out specifically. Everybody who's susceptible is susceptible, everybody who's exposed has been exposed, etc. It's not old versus young, sick versus healthy, things like that. The most common way to do this, the most simple way, I should not say the most common way. The simplest way to do this is with the methodology called ordinary differential equations. I'm not going to go into it, but the idea here is that it's all deterministic. Once one person in your system is infected, your whole system is eventually, over time, everybody's going to get infected. Everybody will recover. So that is, and also there's no chance here. It will happen. Every time you run this kind of model, you're going to get the same result. However, the nice thing is you can use these parameters to actually go back and calculate that R0. So the strengths are, it's a pretty simple approach. It's good for going back and fitting data, trying to figure out what the R0 might have been. The weakness is it doesn't include any chance or randomness. And so I'm going to go to a slightly different version of this. What if we made these events probabilistic? What if there's a chance that an infectious person goes back and infects somebody else, but there's a chance they don't? What if there's a chance somebody who's exposed gets infected, and what if there's a chance they don't? Things like that. And then you start talking about what are called stochastic SEIR models. And these you will see a lot in the news. And they're typically done with these Markov chain Monte Carlo approaches. Again, I'm not going to go into the details of this, but the idea is these are called SEIR or SIR models, and they're stochastic or probabilistic. An example of how this was used early on was looking at how a new epidemic could be seeded by travel from Wuhan to other places. So you have this population Wuhan, susceptible, exposed, infectious, removed, but then you have some of these people, whether susceptible, exposed, infectious, all going to another location with travel. And there they can seed a new epidemic. And after fitting data, this group came to the conclusion, or prediction, it wasn't a conclusion, that their best guess early on was that it would take maybe 3 to 4 people from a different location. Once you have that many, you had a very high likelihood of establishing a new epidemic in a new location. They also were able to estimate that R0 value as rough higher than 2 before travel restrictions and social distancing, and less than that, more like 1 afterwards. So that kind of gives you a sense of how those SEIR models can be used to look at data, say, early on. However, I want to really jump into some of these other models that we've seen in the news a lot. If you recall, about a month ago now, I think almost, there was some modeling that was cited as really prompting the US and the UK to action. So you saw headlines like a report that helped convince Trump to take coronavirus seriously, or White House takes new line after dire report on death toll. This was all based on a quantitative model out of the Imperial College in the UK. Now, this model was very different than the ones we talked about earlier. This one, instead of having these lump populations, they're literally simulating every individual. And how do they do this? They use extensive census data. Where are the schools? Where are the workplaces? Simulate how all these people, like dots on a grid, are moving around, contacting each other, etc. And this is an extremely complicated model. It's called an agent or individual-based model. Now, every time they run it, it's going to turn out a little different because it's probabilistic. Sometimes a person goes to this school, sometimes maybe they don't make it to school, maybe they skip school, I don't know, skip work. Maybe when they meet each other, sometimes they don't. Now, like, as I said, this is a very complex model, and the reason they were able to do this by March of this year, so early in the epidemic, was they were repurposing a model that had previously been developed for influenza for US and UK. And because that model had already been built and taken into account all this complex data, they could quickly revamp it for COVID. So this kind of shows you the complexity of what they're simulating, how people are moving around, when they meet, what the likelihood of them infecting each other when they meet, all of this. And for each individual, they then had a more complicated version of one of these SEIR models where over time, each individual in that big simulation was changing their state whether they go from uninfected to infected to symptomatic to recover. So as you see, this is a very complex model, but the outcome is they were able to really look at the US and UK population very closely. And this was the prediction that really scared everybody early on, appropriately so, was that in the US, you could have something like 2 million people who might pass away or be affected and die by August 2020. Now, the interesting thing is they can not only just simulate what's happening for the disease, they could also go back and model what would happen if we did case isolation. What happens if we voluntarily quarantine or do all of these other social distancing of different sorts, close schools. They could enforce that into their simulations and see how would this change. And now what you see is that, this is Great Britain here, but you see how that curve would change quantitatively from their simulations under all of these conditions, each of these conditions. And this is what really pushed the US and the UK to the social distancing space. But recently, that isn't the model we're using to drive our internal decision-making. And you probably have seen Dr. Deborah Bricks or others presenting this curve on the national stage at the press conferences. And this is from the IHME model coming out of the University of Washington. So what does this model say? And let's think about how it was constructed. Well, this is a statistical model. It's not trying to simulate individuals. It's not trying to simulate populations of individuals. It simply said, you know, if I look at all the death curves from all of the different countries that we've seen data out of so far, it has this shape, this sigmoidal shape, where it starts off steep and over time flattens out. So I'm going, in terms of total death, which corresponds to a rate that looks like this, a very rapidly increasing rate and then a declining rate. Well, I'm going to just assume every country is going to follow that pattern, and I'm going to fit a curve with that shape to the data from each country. And then I can predict from where you are now what your trajectory is. So if maybe you're here, but I could then predict your trajectory, tell you how many we project the death rate at, based on that, kind of back out what we think the medical demand will be, and also maybe hypothesize by changing some of those fitting parameters from their baseline values to, oh, what if I could reduce the increase rate of this by 50% by doing social distancing that's 50% effective. They could make some projections. And this is what you saw when in April 1, they predicted something like 90 to 100,000 fatalities with a very wide error range, confidence interval, by the way. But by April 11th, that prediction had fallen to more like 60,000. And again, the interval is very wide. What, first of all, changed the results? It was the emerging data over time. In that little over a week, the data coming out was reflecting the social distancing that the population had been doing. And so the death rate started changing, and therefore the projections reflected that. Also, it should be pointed out that these confidence intervals do overlap significantly. So, is 90 or 100,000 that different from 60,000 in a model? Perhaps not. In reality, of course, it's different. But I think the point they're trying to make was we're on a very dangerous trajectory. Social distancing, we can reduce it. Hey, we are reducing it. Let's keep it up, etc. That's how this model should be used. But it's important to realize this model can only be used in places where you already have enough of an epidemic going on that you have the minimal death rate for them to start fitting that death rate curve. It cannot be used to assess the very early stages of in a place that's new, which only has like 1 or 2 cases or something. They have a minimal rate that they will only look at data after that. And furthermore, it assumes there are no new seed cases. Nobody enters your community with COVID and then mingles. It's assuming what you started out with is all you have. So that kind of has brought people to say, well, this model is flawed and shouldn't guide US policies. Interesting that people who said these are some of the experts in the other 2 methodologies I've presented. However, I wouldn't go as far as saying it's flawed and shouldn't guide policies. What I'd say is each of these approaches has different strengths. One is better for assessing the data to date and making immediate projections based on trends we've seen other places. The others are far more phenomenological and can kind of be used more for, hey, what if scenarios. So in fact, if you look at a state like California, California does not seem to be relying only on the predictions out of that IHME model. For instance, a recent article suggested that they are using a couple of these SEIR or SIR models also, one out of Stanford and one out of UPenn. Now, those were giving them slightly different results with a later peak. Again, how do you reconcile all this? Maybe the exact moment of the peak isn't, maybe you have to consider that there is some uncertainty in the exact moment of when that peak will happen. Maybe it's best for our governments to be aware that none of these models is 100% predictive and that they have to consider different models. Now, some of these models have themselves been criticized. This bottom one is extremely sensitive to some of your assumptions about the transmissibility. And that could have been what pushed that, that peak so late. What I think is interesting is then saying, well, what do all of these different models or approaches tell us about the next phase, which is what we're all asking about, what happens post-shelter in place. Now, we've seen the director of the CDC saying we need antibody tests in place because it's very likely we'll have a second wave. So let's look at this IHME model. And this was actually what first spurred me to put this presentation together. I had my colleagues asking me, why don't we see a second wave here? Why do they just have everything going away? Because it's a statistical model, and that was how it was built. It was never built for that next wave. It was assuming that the death rate would flatten out over time. and it's only meant to look at the first wave. And so you cannot use this model to predict resurgence. Now we can, however, think about what might this space look like? And I'm sorry for the quality of this graphic. I couldn't find a better version of it. But okay, if we think there might be a second wave, how is that going to look? How is the timing going to be relative to the development of these serology tests, for example, or for expansion of our medical capacity, or for potential vaccines, things like that? Well, some of these other models have looked at that. So the ABM model I talked about, in fact, it did simulate what would happen if you lifted social distancing at this point here in their model, September 2020, and how there very well could be a second wave. They predicted there would be a second wave, in fact. And if you look close up at this data, different mitigation strategies would affect the size of that second wave. They also simulated, well, what can we do if we start seeing these second waves? And they did what they call on-off social distancing, where when it starts to happen again, we go back to social distancing, then we relax, then we go back. Well, that's great and all, but I'm not sure how pragmatic that is as a governmental policy to suddenly every few months announce, go back to your homes for another month. That would probably really cause a lot of chaos in our communities. So maybe what we will be doing is combining or layering containment strategies, and this is what we've talked about, a slow return. But before I get to that, I'll point out that other models have also looked at this. This is the one that California was supposedly looking at, the Stanford model. Again, they called this light switch social distancing or light switch maintenance. And here, it's interesting to note that while they call it light switch, Newsom, who's supposedly looking at this model, made the comment, there is no light switch here. It's more like a dimmer. So we don't necessarily want to turn on the light, turn off the light, turn on the light, turn off the light. Can we dim it? Can we get to an intermediate stage where we can reduce the transmission and keep things manageable until we have either herd immunity or a vaccine? And I thought this is, I'd just end with this very recent publication. It came out maybe sometime this week out of Harvard, which takes that same concept of projecting with these SIR or agent-based models. Here it's an SIR model. And they say, well, they even considered things like, what about seasonality? Or what about increased care capacity? So from looking at other past viruses, SARS, et cetera, they said, well, if this has a seasonality like that, and we layered that into our model, and if we can simulate increased care capacity over time, what might this look like? And they even can include that in trying to help our governments think about what's needed and what would be most impactful in terms of influencing the spread of this disease and the hospitalization associated with it. And ultimately, what we're really talking about is in the schematic that many of you may have seen from Thomas Poya, where he talks about an immediate hard hit, where this is where we are right now in this hammer stage, where we're really just like strict social distancing, to what he calls the dance, where we're going to do some kind of intermediate solution, not light switch, but maybe some of the restrictions and not all of the restrictions, et cetera, to kind of just keep things roiling along until we can get, and maybe that's where testing can help, right? And in these models, you can actually implement what happens if you test, what happens if you quarantine, what happens if you isolate. So it can really help us determine what are the most promising strategies to keeping that curve low while we wait for a vaccine. So with that, I'd like to also point you to a few other general good reads on modeling. Again, you don't need to be an expert in modeling to understand any of these. They're very descriptive. And for those who are more interested in actually using simulators or trying them out, there's a few online tools. So with that, I will end my presentation. And thank you, Amita, for the invitation. And I will stop sharing.
Thank you, Saroja. That's great. Amita, can we go back to the agenda slide? Just leave it up there so that although we don't need a slide up
We
there.
don't need a slide. We don't need a slide. I want to see
Just see it
faces.
on the panel view.
Yeah,
Actually,
don't put the slide. Yeah, unshare.
because
I
then
want to see
you
the
can't.
faces.
I agree. I agree. So let's just do the panel view. This is awesome. And everyone, feel free to turn on your videos, because since we are on a panel view, we can see each other, and it's a small group. We don't have hundreds of people. So, if you don't mind, if you could turn on your videos, that would be wonderful. Great discussion. Great presentations by both Sanjeev and Saroja. I've been a voracious reader of stuff that's coming out on this topic, involved with a platform called coviddb.org, where a lot of information is being put out. But frankly, I learned a lot of new things from both Sanjeev and Saroja's presentation. Pretty deep stuff there. But there were a lot of really good questions that were coming out, and we'll go through those as we go through this next 20 minutes. We're a short time, and we may not be able to go through all the questions. So I requested Sanjeev to start responding on the chat window, and Saroja, you may do that as well if you feel like it right now. But let me ask you a very provocative question to both of you, Sanjeev and Saroja, and if anybody else wants to weigh in, can weigh in. It's since the beginning of this crisis, whether you call it December the start point or January the start point, there's a lot that we have learned about this that we didn't know the day before or the week before. And as we are moving forward, there are new things that are being figured out all the time. And it's not just the cumulative data that's providing more resolution into what kind of people are being impacted and what sort of complications that are coming out. That's definitely part of it. But even virologically or serologically, there's a lot that we don't know. Can I ask both of you to conjecture as to what are the things that we don't know today about this virus and how it behaves, that we would be learning more about in the coming weeks and months? What kind of things? For example, there's a question about reinfection. There's a question about having antibodies that may not create a immunity. Things of that sort. Could you talk about that? Maybe Sanjeev, you could go first.
Yeah. Ishita, I think Ishita just asked that question about biomarker and what is a disease. Yesterday, that paper that was published, I have the citation on my slide, said, they talk about this COVID-19 is a completely different disease. What they are seeing is that patients with asthma don't show up. They don't get infected. Normally, you would think that they are the ones with the weakest lungs. This is a lung disease. Typically what happens is a lung thing. They are the ones that get infected first. They don't see it as much. This seems like more like a metabolic disease, where the ones that are diabetic, having metabolic issues are showing up. There are and that slide that I had where you're seeing heart attacks, heart-related events, intestinal, kidney, pancreatic, and they're not going into intubations at all. They're just dying because of a completely different organ failure.
Can I ask two quick follow-up questions to that, Sanjeev? So there's a physician in New York, without any sort of real study, because he didn't have the research tools to study it, but he was saying that a lot of patients that he's seeing that are dying, they are potentially dying because of the interventions that they are performing in the hospital, so such as intubation and sort of more invasive form of ventilation. Could you talk about that in the context of what you just said?
Yeah, see, that is even a bigger question, because whether to intubate or not is a decision that the intensivist makes. It's a call that person makes, and clearly we are seeing 50% fatality intubated because of the intubation or because of the disease. There is septic events. So no, it's very hard to say. At this point, a lot of it is unknown. So whether to—
Can I make a comment?
Yeah, go ahead.
Ramani, you have a question?
Yeah, I want to just follow up on this discussion about the respiratory, the aspect that Anurag brought up. And I think I saw the same video from this doctor in New York. And what he was mentioning was that the conventional approach to treating this has been acute respiratory distress syndrome.
Correct.
In which the ventilator is set up to assist the muscle to pump more of the patient's own oxygen. And he felt that the symptoms that he was observing were more akin to what he called altitude sickness, where it's more of a lack of oxygen rather than a muscle problem, in which case the treatment approach has to be different. And so the ventilator settings have to be different and some changes are needed. So I wonder if, I don't think we have necessarily a doctor on our call, but anybody can comment on those kinds of issues being addressed.
So I think he was talking about ARDS protocol. So most of the hospitals, and my nephew just went through this at SF General, so I researched that a little bit more. He was talking about ARDS protocol, and that is on the pressure control mode. And I think we don't have a ventilator expert on the call to talk about that, but I think he was talking about a different mode of using ventilators. So pressure control versus maybe volume control or some other.
So Darshana just sent that article. That's the article that Darshana, thanks. That's the one that I was looking at. And we are going to learn a lot more. And everything that you're saying is correct, this is not just ARDS. You do see low oxygen levels that cannot be easily explained why this is happening. It's not just pushing oxygen in using a ventilator. So only time will tell.
So Saroja, do you want to weigh in on that question? What are the things that we don't know today that we will learn?
Well, I think it ties very much back into what we were just hearing. And that is, as we learn more about what are the risk factors, what is actually causing the death, etc. While those seem like biological insights, they're also population insights. So in any of these kinds of modeling approaches, you need to understand how to bucket your population. What is really the risk? If you're going to be in a Michigan or a New York or a Detroit or a New York City where you have a large African-American population and you have pre-existing obesity, diabetes, and just generally inequity in healthcare through the years, then you have to revise the way you even statistically project what can happen to that population, or those fundamentally change the death rates that you may see, or the severity rates of infection that you will see in those places, rather than a San Diego or where you have a more a very different population, for example. In addition, I mentioned how, as we learn about the medical expansion, how do we expand medical care, etc. Maybe it's not just expansion of the system. Maybe it's not pure volume, but maybe it's how we manage, and maybe that leads to a faster turnaround and a faster exit from the hospital for patients who recover. And of course, the development of vaccines, the understanding of the infectivity at different stages, in the pre-symptomatic stage or the post-symptomatic stage, at the understanding of the durability of immunity that somebody may achieve after having been affected. That's all going to impact what we project will happen in the future.
Saroja, may I make a comment?
Mm-hmm.
I wanted to really thank you for the really nice overview on the projections, because I've been very confused about what people have been talking about it until you put it together. So it's just good to know, and the resources. The one thing that people don't seem to be addressing is there is a static picture of what happened initially with California doing extreme measures of social distancing. We're all following rigorously. And then New York got onto the act about a few weeks later. They had a discussion flight between Cuomo and de Blasio and all that.
Yep.
Then we have the whole spectrum of places like Florida and all these red states that are reluctantly following something, not completely. Even yesterday, he's opened up the beaches, for instance. And then we have completely eight states that don't even want to do anything about it. There's total, So how do you model these things in? Because I'm sure there's going to be a second wave. The question is, when is that going to hit? Based on, I don't know if any kinetic
Can I add to what Ramani just asked? So is there going to be, so everybody's talking about second wave, but you also presented one of your slides, Saroja, about this being a tap that turns on and off, or a language like that. So it may actually be ripples that are many-fold rather than just a second wave. Could you weave that into your answer? And then we'll move on to the next question, which I'll channel to Sanjeev. Go ahead, Troy.
Sure. So first of all, in some of those models, they did look at things like how would a 2-week or a 1-week delay in implementing these strategies affect the death rate, and it was marked, or the hospital utility. It was marked. You're talking about an exponentially growing scenario. So a week is a huge 2 weeks is a huge amount of time when you're talking about that number of people. So they have I don't think they explicitly looked at New York versus California, given their initial conditions, their population density, et cetera. But for a given setup, a given location, 1 to 2 weeks is a huge amount of time to delay social distancing. They also have explicitly looked at things like what is the extent of compliance with social distancing. And they have made different projections. For one of the resources I have on the last slide actually says, what if you have very good compliance? What if you have poor compliance? And they show projections under those different scenarios. And even one of them looks at what if we put a compliance fatigue in, where for any length of social distancing, it starts out good. And after the first month, people start cheating because it's just like a diet, right? And they look at that too. And that can all be if we can really get this in place, and if we have an administration or administrations in the different states that want to look into this, they really can look into this with the computational models and try and make, at the very least, qualitative predictions about the impact of these different strategies. Is it really critical that we act now, or can we wait 3 days to see how things happen, or is that too late? And in terms of the timing of that wave, they do predict the timing of that wave based on when the social distancing is stopped and based on different strategies. But you have to also consider, one of the critical things is that they talk about case quarantine. And imagine if every time somebody tests, let's say you really could test everybody, and you know that your population, which has been closed, is past its wave. But now, if you're testing, if you could take every person who is even asymptomatic positive and put them in a bubble and just quarantine them for 3-4 weeks, theoretically, you would never get a second wave, because every time somebody showed up, they would be bubbled. But how pragmatic is that? What does that require in terms of testing? So yes, you can explore those things. Yes, retroactively, we should, while we look towards a second wave or a future pandemic even. But the explicit details have to be to that location. Absolutely. There's one last comment I could make is we also have to look at how much interconnection or movement or exchange of people occurs between these different sites. I can say I don't really care what happens in Florida. If they're going to do that, fine. It's their problem. Except, guess what? Florida doesn't stay in Florida. Well, I do care what happens in Florida. There are people there. But even if I didn't, Florida doesn't stay in Florida. California doesn't stay in California. We're not going to be checking and shutting down all travel indefinitely. So we have to think about the pragmatics of this whole nationwide and worldwide dynamic.
Absolutely. So the great answer. So the next question, I'll go through some of the questions that are on the chat window. People have been patiently waiting for those. I think Sanjeev has responded to some of them. I've responded to some of them, and we'll follow up on others that we can't get to on this short Q&A session. But the next one is an interesting one because everybody is concerned about that, and there's a lot of uncertainty around it. And I'll bring that question to you, Sanjeev, because remember microhydrodynamics of surfaces course that we did it in Colorado, that's directly related to that. So the question is from Ravi. It talks about virus being airborne. How does it work? I mean, early on we were told that this is not airborne, that it actually, people don't need to wear masks because frankly, you can't really transmit it that way. It's really people who are infected who made projectiling, a spit or something that, a phlegm or something that could actually carry that, but that's the only way. So how does it work, and does it stay in the vicinity of the person who coughed or spoke or traveled farther? Or if you're just in, you're jogging behind somebody 2 or 3 or 4 or 6 feet, and somebody's just kind of, is breathing heavily, and can that actually infect? So can you talk a little bit about the hydrodynamics of that, and how does that work?
Yeah, it was initially everyone thought that during incubation in these procedures, that's when these particles are generated. but there are now papers in Science and Nature, as well as German papers that did simulations in especially closed environments. So if there is no running air in the room, and if somebody sneezes or coughs, not only the distance to which their droplets travel, but then micro droplets are formed that could stay around for a couple of hours later even. And that was what this article had shown. Subsequent to that, there were multiple other articles that talked about the efficacy of N95, N99 versus cloth masks, surgical masks. And now it is very clear. I think Contra Costa or Bay Area is already requiring face coverings. My problem is that they're not as available now still as widely. I would like them to be available as chewing gum on any Walgreens and Targets. And I think we will have to wear, especially in closed spaces, keep social distancing. But I clearly it is transmitted. The respiratory is the way it is transmitted. And if you are in that room, then any person that has any semblance of onset of symptoms, respiratory symptoms, should isolate themselves. Yeah. I mean, in hindsight, it seems like simple in 2020, but micro droplets carrying has been known for a long time, as you know.
Yeah.
I'll send that article to Rob Davis, our advisor from Boulder.
That's right.
If I can clarify that quickly, let's say I'm walking in my neighborhood. Somebody just walked before me about 5 minutes back or 10 minutes back and had a cough or sneeze, and I'm just going through the same stretch. And if I don't have a mask, what happens to me?
So in Belgium, there was an article that showed that, the 6 feet doesn't work if you are actually running behind someone and things like that. You have to, in my opinion, you have to use common sense. If somebody, first of all, in an outside environment, you talked about beach, right? Things are going to be carried away by breezes. Yeah. So utilize your judgment call. It's not like they're not contagious to the extent they're just going to find themselves and into your lungs. There's a certain viral load that person must be carrying. That viral load has to find way into your respiratory passage. You have to touch it. So, I mean, I don't think there is a clear.
Yeah, and my thought is that if you really want to be safe and you are a vulnerable population at a certain age or certain comorbidities, you should exercise all of the precautions. Yeah. Because it's a game of odds, and I think you talk about probability in there, Saroja. Absolutely. I mean, there are a lot of it's not a monodisperse population that we are talking about. So the same conditions may impact one person versus the other differently. So my recommendation would be to use personal protection if you're concerned about that. Let me move on to another question. And I'm just picking certain questions, and there may be others that I think we have responded to, but we may get to. So there's a question about what factors explain the severity of infection and mortality in different regions and countries, China versus Italy versus Singapore versus Germany versus here. And I had a response in there saying, I said about the population we are talking about, comorbidities that exist, the infrastructure that country has, the compliance that we have in terms of social distancing. But there's another thing that I just heard on a School of Medicine call last week about these strains. Apparently the Bay Area has 7 or 8 strains that are different from the strains that were seen in New York. New York strains came mainly from European route, from Italy and other places, whereas the one that we have here are more from Asia. And somehow the conjecture is that the infectiousness, the R0 rate for the virus here, and R0 is not sacrosanct, so is CFR. CFR is also not sacrosanct, these change. But underlying biological, the virology of the strains, it seems like that's different between these 2 regions, for example, New York and San Francisco. Can you both, Sanjeev and Saroja, can you talk about what explains the differences in the severity and the mortality that we are seeing in these different regions?
I can give you my comment.
Who said go ahead?
No, I think, there was an article comparing really what happened in Asia versus Europe versus US, looking at the time at which people, everybody blamed China, but once that happened, Taiwan and South Korea had very different experiences, and to some extent Germany compared to Italy And there's also head-to-head comparison of Sweden versus Norway, where the fundamental difference appears to be the rate at which they started testing very early on and started doing contact distancing and all this kind of tracing to put people aside. So it was more than a strain problem biologically. It was really a public health solution that actually made the difference early on. I think South Korea and Taiwan in particular, after the SARS experience, they really overreacted in such a manner in which they had a testing in place in a big hurry. And in fact, South Korea and US both found out about the first incidence was in the same day, but the reactions were dramatically different. We had a situation where people are calling it a hoax, and there they went into the other extreme and put in all these testing and other such things in place. So Anurag, your point is very well taken about different kind of strains that has to be addressed. But I think early on, the differences appear to be really fundamentally based on the public health policy approaches that made a difference. I think that's my 2 cents.
But I do want to hear Saroja and Sanjeev's thoughts on that. Saroja, do you want to go first?
Is it, you're talking about the different incidents and spreading?
Differences in severity and the mortality. And Ramani just brought up Sweden. Sweden is now experiencing a pretty rapid increase, and they had not followed any of the protocols that we followed here. For example, Singapore is going through a lockdown now. Japan, I was in Japan in February, early February, and there was no. I was in a packed bus when I traveled there to one of the most affected places, Hokkaido region. So they're suffering now. So I agree with your points, Ramani, that a lot of that is about that. But is there a biological reason also? Are there different kinds of strains? So
I do think there is a fundamental difference beyond just the social policy. I do think the social policy and the government policy is a huge factor, okay? And people's following of that, etc. But I think you can't look at the numbers that are coming out on the severity rate, the death rate in African Americans and just brush that off. And it seems to be, I think we have to look more fundamentally. Is it some difference, some biological, maybe, but it right now seems to track with overall health status ahead of time. So were you generally in good health? Were you obese? Did you have chronic lung issues? Did you have chronic any health disorders that are of the sort that are making for the worst case scenarios here? What is the time period before different people even get care or go for care? People are avoiding hospitals now. I mean, literally, many of the hospitals have reported people are coming in with cardiovascular symptoms post-stroke by days, post-heart attack. This was on the news 2 days ago.
70% decline in number of stroke treatments in the hospitals. 70%. Stroke is an emergent condition, and people are not showing up at the hospital even after they have stroke and heart attack.
And when they are later.
Yeah.
It turns out they already are well past the ideal treatment window. But my point was that people are also avoiding care and may not have easy access to care. And if you don't have insurance, even if the government says, we won't charge you for this, there is a lack of familiarity, a custom habit of going to the doctor. I think all of this is playing in. And are these the people who are in the essential jobs? Are they the people who are the least protected? I really don't think we know all of this, but there is something undeniable beyond the.
Can I ask a follow-up question? Can I ask a very quick follow-up, because this relates to India, which is a country we all care about. So if there's, if a primary factor, one of the primary factors is, health status of people and our ability to deal with that, then is India at a significant risk then compared to some other more developed healthcare systems where health status is presumably better at a population level? And then Amita, maybe you can ask your question.
I'll just quickly say it depends. We still don't know what are those things that make the person at most risk. If it's obesity, maybe not in the same population. If it's general health, maybe so. It's until we really have a good handle on why certain subpopulations, etc., are having such bad outcomes compared to others. It's hard to project, because under some of those circumstances, India could be way worse off. Under some of them, they might be okay. Who knows?
Yeah.
You had a question, but I'm happy to chat later. Yeah, Amita.
Yeah.
Yeah, I wanted to share, I just shared a website that has
Yes,
Next
yes,
Train Team website that
yes,
really shows nice phylogeny of the coronavirus and the
yes,
genomic
yes.
epidemiology. Haven't dug in a lot, but it is very clear, you know, like the New York, what happened in New York versus what happened in San Francisco. There were different mutations in the virus.
But, but but I looked at it, and the phylogeny tracks from the first patient in early December to now. They're tracking.
Yeah.
But what is very interesting is all these mutations are not in those 120 amino acids where it's
Exactly.
the spike
Exactly.
protein that is binding.
Yeah, so it hasn't evolved like HIV where you have type 1
Exactly.
and two different types. So it's still the same, and it still is infective. It hasn't yet evolved
Correct.
to a place where it's
The
yes.
Yes, mutations are the ones where influenza mutations every year CDC has 3 different strains, right, in the flu vaccine.
Mm-hmm.
Or there are other mutating viruses. HIV is one of them. It's hard to make a vaccine. This is unlike it. This is actually much smaller than the SARS-CoV-2 from a length standpoint.
Mm-hmm.
General consensus, what I've read of the folks, is that it is unlikely to rapidly mutate. And my opinion of the differences are clearly in one, the culture, whether it is distancing, whether it is obeying the government versus, in China, if they say wear mask, every person will wear a mask, they anyway wear masks. They're not going to question it. Whereas here, individualism, so that balance of individualism versus community, that cultural contact tracing. I mean, Germany had corona taxis. So they had actually corona taxis where their taxis would go to the place, check them if somebody felt like they were exposed. If they're positive, they were asked to isolate, they were monitoring. I mean, and that's German, it's a stereotypical German management. And that is why, so there's one thing about incidence and second about fatality. And German fatality is very, very low because of their excellent management. I think we are also going to learn a lot from what is happening in New York, all this thing about intubations and what is the right way to manage these patients. Our fatalities are going to come down, definitely. There's no doubt about it. I've got tremendous confidence. Infectious, if we follow these, even if we open the beaches, but if we follow, in my opinion, face masking, keep a distance, I think it will still be okay, in my opinion. Not immediately, but within a month or whatever the models indicate.
So what
Can
I'm
I make a
hearing
quick
here,
comment about
go ahead.
about the strains, about the the different kinds of. I think being an RNA virus like HIV and others, there's a likelihood that there'll be a lot of mutant strains around, but whether they'll be clinically relevant or not and have an impact,
Exactly.
we will know only after vaccine comes into play. Because that's when you'll really know how effective the vaccines are, and seasonally over a period of time is when we learn that. Right now, it's very early to sort of.
And if it puts a evolutionary pressure. I'll say one more thing about the care. One really good example is the move towards proning patients, which is putting them on their stomach.
Yes.
It seems to be, many doctors are saying it is extremely effective. It's the last step measure before they intubate or anything. If we prone them, many of those patients avoid having to go to intubation. Is that anecdotal? Is that the stats? How is it holding up? We'll see. But that may be a very simple management approach.
Yeah. It's more than anecdotal now, Saroja. There are now papers that talk about proning.
Yeah. Well, I know we have a lot of really interesting questions, and we could actually go longer. We only had 20 minutes for the Q&A. So why don't we stop here, because we are eating into Robert's time to talk about some exciting things that are coming. Robert, I'll hand it over to you.
Okay, perfect. Can everyone hear me okay? Can see my slides?
We do.
All right, great. All right, so we're going to switch gears here for the last few minutes, folks. And thanks for everyone joining us. We have 34 people online, which is great to see given the kind of 24-hour notice we provided here. So for the last few minutes, I want to provide some update here on EPPIC overall, what we've been up to, and what our plans are in the near term, and how you can get involved, right? So gonna just cover off 3 slides. So we're gonna try and keep this last portion here to only about 10 or 15 minutes at the most. But really the 3 topics I wanna cover with everyone are our strategic framework. We spent a lot of time on this over the last period of months to really pull this together and get it fully aligned across the group. We'll then spend a few minutes talking about our value model, all right? Who should be part of EPPIC? What are the different membership categories, and what are we offering? And then finally, how do you become a member? So, strategic framework, all right? So we spent, like I mentioned, a good bit of time, and I think for folks who've been involved in EPPIC, which are most people on this call, I mean, this is really no surprise. At the same time, you know, I think to document this in one place and be able to share it like we are now is something relatively new. So I want to spend some time walking through this with everyone on the call. So firstly, our goal. Our goal is to be a leading forum for healthcare and life science professionals in the regions it serves, right? I think pretty straightforward, and I think for those who've been involved in EPPIC, that's been a consistent goal for us. And similarly, our strategic positioning. We talk a lot about networking, mentorship, and entrepreneurial support. So we say that our strategic position is to be a premium forum for networking, mentorship, and entrepreneurial support of professionals in the healthcare and life science industry. Okay, and we also mention and pay justice to the fact that we are specifically angling towards folks who are in or have a connection with the U.S.-India corridor. Now, certainly that's not to say that we are only targeting U.S.-Indian type of content or events, but that is a part of our demographics, a part of our history, and we acknowledge that. But on top of that, we certainly are targeting, more broadly speaking, this intersection between technology and healthcare as it relates to serving mentorship, networking, and entrepreneurial support. So how do we do that? How do we enable these goals to happen, and how do we maintain this strategic positioning? So this really starts with our member base. And so what we describe here as our core pillar is growing our membership fundamentally. And this speaks to the fact that we need to understand who our members are. We need to understand what is it we're providing to our members, and we need to build a robust strategy around how we're going to continue to grow that member base. And so the strategy that we have is really, I would say, summarized in these 5 enabling pillars. Okay, so these 5 enabling pillars we'll walk through here briefly. And the idea here is that each of these pillars provide one portion of what collectively will help us to grow our member base and ultimately achieve our goal. So the first pillar on the far left talks about developing a high caliber, diverse leadership. Okay, that's really an internal capability, refers to when we have members, when we have people who are part of our member base, how do we make sure that there's a pipeline there? How do we make sure those folks are becoming leaders within our organization that can be basically a funnel into both the EC as well as the board, as well as into the broader community that we serve. We then talk about having a sustained value-focused programming, all right? So we've seen this in the past. That's things like all the events we hold, EPICon, quarterly events, et cetera. How do we maintain that programming in a way that's holding true and holding its value? And value being both in terms of the value that we provide to our members being realized, as well as staying true to our own values in terms of the type of programming. We then talk about our partnerships, which is our third column, which is really making sure we engage with other organizations, right? I mean, I think our ability to execute is partially limited by how many folks we're able to pull in and leverage. And so how do we keep our partnerships alive? That's partnerships like our corporations we work with, other similar nonprofit organizations, as well as academia. And there's a number of partnerships outside of that as well that we're looking to potentially add on to. We then also talk about our fourth pillar, which is messaging and thought leadership. And that's really, I would say, something that we're taking a very active stance on right now. And that's actually what we're doing at present, right? It's, how do we create thought leadership and messaging that's outside of our typical forums in terms of our in-person events? So that's going to be things like webinars, and today's a great example of that. How do we expand over and above that? How do we create more content through electronic communication, through, say, our website or through social media? And then finally, financial sustainability. So we need to continue to improve our internal systems and practices around maintaining our finances. So This is an action-packed slide, which really summarizes a lot of the discussion we've had internally, and a lot of what you've probably seen over the last number of years. But we're really now structuring ourselves very closely to this, and so we're building an internal organizational structure to support this vision and coming up with clear goals in line with this. And so that's probably as much detail as I'm going to go into in this presentation, given we only have a few minutes here. But I just want to provide this kind of all in one place, and I'm happy to answer any questions on this towards the back end of this, as well as offline as we get more interest. But I do want to then transition here and talk then, okay, we have this strategic framework, but what are we actually providing, right? And that's where we talk about our value model. So we have to be clear about what our value truly is with EPPIC, all right? And it really is us. And what I mean by that, it's the inherent value that we bring. I mean, there's 34 of us here who decided it was a good usage of our time on a Sunday to call into this webinar and contribute to this conversation. And it's that type of robust sense of community and sense of interest in healthcare space, which has kept EPPIC around, and it's kept EPPIC relevant and robust. So it's really our ability to mentor a wide range of professionals through our own successes that we've had collectively. It's also our deep passion for healthcare, as well as our expertise in a diverse set of healthcare domains. So we're really looking to leverage this value and provide that to our member base. And through that, we're creating a bit of a different model than we've used historically. So firstly, we're taking an active stance now to actually establish clear different membership levels, okay? So historically, we've only had the charter membership level clearly defined, whereby after that, his membership has typically been defined as those who sign up for and attend our events like EPPICon. Moving forward, we're trying to really create a more member-based model as opposed to an event-based model. And so what we're doing here is defining different membership categories that will look to drive programming content and different types of content to be very specifically angled at, to create value within these different buckets. So there's 4 categories, and actually 5 categories, right? So on an individual level, individuals can join as a charter member at a rate of $500 per year, an entrepreneur member at $200 per year, a professional member and a student member at $150 per year respectively. Finally, also we acknowledge our corporate members, who are also a key part of this model. So who are we looking to join, right? And it's a pretty, we're not excluding too many folks here, but to just put it down, look, I mean, entrepreneurs are for sure part of this community. Many of us on this call are entrepreneurs, and that's one of the biggest, most robust communities that we serve. But also it's many professionals in healthcare and many professionals who are not in healthcare. So folks who are not in healthcare but want to get into healthcare to have access to our network, for example, as well as folks in healthcare who want to have either influence or seeking broader connection base. In general, we would seek to have these folks be the type of folks who would fall under these charter and entrepreneur categories, whereby we would also look to have, high school, university students, as well as entry and mid-level professionals join us within these membership categories. We are looking to create content that would be specific to those, value offerings that are specific to those different member types. And then finally, corporations. And I would say we're still in the process of driving our value statements as far as corporations are concerned. I would say we've developed clear value models for corporations that sponsor us as part of our events, but we're also exploring the potential to create, say, corporate members that are more ongoing affiliations with our group. So what are we actually offering, right? And that's the key question here. And there's a lot more detail behind this, but in a nutshell, it's really network access, mentorship, and specialized domain knowledge. So across these 3 areas, we're in the process, and we've already identified a number of unique value offerings across these 4 member type areas within these kind of specific offering types. And so we're in the process of doing more to define this, and we have a lot of conversation happening internally at the moment. But at a high level, these are the 3 areas that we're really focusing on, and we'll look to achieve those through a combination of in-person events, as well as different types of content to be distributed online. So finally, and this is my last slide for everyone, how do you become part of EPPIC, all right? So firstly, we're aiming for significant growth. So we are changing our value model, we're changing our strategic strategy, and we're really getting more defined around that. So To join today, what would you get? You're going to get a much more in-depth focus on, or look on what I just brought you there, right? So a more enhanced visibility to what we're actually doing internally. We're looking to get people to have firsthand ownership within our organization. So you can see on the right-hand side, that's a picture of our organizational structure, which is a direct map of effectively our strategic plan, and really be part of the movement. I think we're at a formative stage right now where we're looking to really pivot and really broaden our impact and really structure our member base. And so being part today gives you insight into how we're taking that off the ground. So one question is, what do we plan to do in the next year? All right, and it's really in this order. So where we are today is we're really actively setting up our communications and our finance platforms. That's through primarily our website as well as social media platforms. Once those are set up, we'll launch, all right? And through that launch, we'll enable content on a variety of platforms, which will really kick off our formal recruitment phase, where we'll look to really angle at driving recruitment across those different membership categories. And we look to get to that phase over the next couple of months. We'll then look to apply those learnings and scales. As we move into the 2021, we certainly look to ramp up and really scale this to be more broad-basing electronically, and then see how we can convert that into bigger in-person events, clearly also contingent on where COVID-19 goes and our ability to host those in-person events. Another question might be, how would I be able to get involved once I am a member? Okay, so as you can see, we're creating different types of teams internally. What I would say at a high level is you can either be involved in strategizing, so strategizing in terms of how we drive our value and how we're growing our organization, as well as execution. So execution on helping us build these systems and develop our content. And the focus within those areas is really through those 5 pillars, which you have on the right-hand side, which are summarized as leadership, programming, partnerships, communications, and finance. All right, so finally, sounds great, right? So what's next? So right now we are accepting accepting applications for charter members. I have provided a link here, and we'll provide that link out through email following this event. As mentioned, we're looking for $500 for a 1-year membership as being a charter member. Okay, and so that's really it. So I just want to say thank you very much, and I'm sure Sanjeev, Amita, Anurag, feel free to chime in, but it was great to have everyone join us on such a short notice. I wanted to show here just on the right-hand side, these are the current board members in blue, as well as our EC members in orange. So we have a great crew that's fully mobilized and rallying to achieve our goals. And we're looking forward to hopefully having some charter members joining us soon. So thank you very much.
This has been wonderful, wonderful to have everybody join. Thank you so much. And thank you to Saroja, to Sanjeev, and to Robert for preparing, and also Anurag for moderating our session. This was really helpful. I think we made. I think this is the power of this organization, that we can get together like this and have discussions. So I've been hearing getting a lot of texts and also watching the questions here. People want to have these kinds of sessions and content like this, and so we would love to get more ideas from you, and we would like to see how we can do this more. But we need our members to contribute and participate. That's the only way we can do this. It's a self-run organization. So love for your contributions, and we would hope that you can become a member, any kind of member, that is charter membership. But Robert also showed, if you go to the previous slides, different levels of membership, and we are planning to show, to provide different kinds of content as well, and host different things for the different kind of membership. So hopefully we'll continue to grow the EPPIC family and provide meaningful things for you.